Hello, everyone. Thank you for joining us today. We are going to give our fellow attendees a few more minutes to come into the webinar, so please continue to hold. We will start in a few minutes. Thank you, everyone. A very good morning, afternoon to you all, and a warm welcome to our Nature Review Bioengineering webinar, where we will discuss the real-world impact of bioengineering and the role of the new journal in supporting scientists at all career stages with their research. Thank you all very much for choosing to be with us. For this webinar, my name is Fariel Zubair. I'm the Customer Engagement Events Manager at Springer Nature, and I'll be hosting today's event. First of all, it gives me a great honor to introduce you to our speakers and support team for today. So here they are joining me live. Christine Horejs, she's Chief Editor, Nature Review Bioengineering, part of Springer Nature. Robert Langer, David H. Koch Institute Professor at Massachusetts Institute of Technology. Annie Moisan, Program Director at Wellcome Leap, Human Organ Psychology and Engineering. Hello, Annie. Nesma Al-Said Ibrahim, she's Associate Editor, Nature Review Bioengineering, part of Springer Nature. Unfortunately, Nesma is not joining because of some other commitments, but we have with us Sadra Bakshanta. He is also Associate Editor, Nature Review Bioengineering, part of Springer Nature. And finally, by no end, by means, I have by support my behind from Esther Morrison. She is Marketing Manager, Research, Marketing Planning at Springer Nature. Okay, now let's have a quick recap of the agenda today. Christine will be giving a brief introduction about Nature Review Bioengineering. Then we will have a panel discussion with our guests today. And at the end, we will have a Q&A session with our audience and our panelists. Before we start the presentation today, I would like to draw your attention for a few housekeeping notices. Today's webinar will be recorded, the link to which will be sent to you shortly after the event. All attendees are in listen-only mode, so please send us any questions you have using the question box in the control panel at any time, and we will answer them at the end of this session. Should you be dropped out of the webinar at any point, please use the same login details to log back in when you are ready. In order to help improve and enhance future sessions for everyone, we would very much appreciate your feedback. Therefore, at the close of the session, you will be directed to a short feedback form, which will take a couple of minutes, but helpful for us for our future events. So here we hope very much that you will get a lot out of this webinar. And without further ado, it's my honor to hand it over to Christine for the Nature Review Bioengineering introduction. Over to you, Christine. Hi.
Thanks so much for the introduction. I'm Christine. I'm the Chief Editor of Nature Reviews Bioengineering, and I want to welcome you all today to this webinar on the real-world impact of bioengineering. I'm delighted to host the discussion today with Bob and Annie and to introduce our new journal, which we will launch very soon in January 2023, Nature Reviews Bioengineering. Let me see if I can progress the slide. Yes. So bioengineering is a very broad field and it's inherently applied. After all, engineering is the science of problem solving. So bioengineers leverage fundamental knowledge to really design and optimize and engineer solutions that work in the real world. Bioengineering has really enabled key breakthroughs in biomedicine. For example, the COVID-19 mRNA vaccines, which we all know so well now, are based on a bioengineered technology, that is the design of lipid nanoparticles to deliver nucleic acids into our body. Similarly, immunotherapy, which perhaps is one of the biggest advances in clinical oncology in the past decades, requires the engineering of immune cells and the precise delivery of immune cells, which has also been enabled by bioengineered technologies. Bioengineers have also developed very important in vitro platforms, such as organs-on-chips and organoids, that allow us to study human health and disease and physiology in vitro without requiring animals. And these models may well bridge the gap between preclinical animal models and clinical trials. Bioengineering also plays an important role in global health related issues, because bioengineers are developing point of care diagnostic tests that can work in a variety of settings, and importantly at the point of care. On the more sophisticated end of the scale, bioengineers are also exploring new ways of monitoring human health and disease, for example, by interfacing bioelectronics and bioelectronic devices, and also robotics and brain-machine interfaces to really assist patients and to really have an immediate and positive impact on the life of patients. And beyond biomedicine, bioengineering also plays an important role in issues related to sustainability and climate change. For example, bioengineers have teamed up with synthetic biologists to engineer microorganisms and plants in a variety of applications, including the plant-based manufacturing, for example, of biopharmaceuticals or for crop improvement. So bioengineering is very broad, is very applied, and is a very exciting research field. So it's about time that our field gets their own Nature Reviews journal. So Nature Reviews Bioengineering, we want to publish authoritative, accessible, and agenda-setting reviews, perspective, and comments covering the full breadth of bioengineering. But important really is the focus on application, translation, and technology transfer. So considerations about product development, about funding, about policy, about regulatory issues are important aspects of our articles. Broadly, we're going to cover 3 different areas: biomedicine and health, biology and environment, and devices and technology. And of course, these areas are fluid and dynamic, as is the field itself, but really everything from more traditional tissue engineering to artificial organs and vaccines, delivery, nanomedicine, to synthetic biology, plant biotechnology, applied microbiology, and protein engineering, to really hardcore device development, bioelectronics, microfluidics, chip technologies, robotics, and wearable devices. So our scope is very broad. I mentioned already the importance of application and translation. So we'll encourage our authors to really discuss low resource considerations. So how could this technology or system or device function in a resource limiting setting, such as a lack of a cold chain or a very distinct environmental conditions? And of course, how could it be translated? What are the issues related to translation, scale up, manufacturing? What about industry collaboration? So we want our articles to really very honestly and frankly discuss how this specific bioengineering research can make an impact in the real world. And we also want to be an inclusive journal. So we are encouraging all our authors to include a citation diversity statement in the review articles to really reference relevant papers in a manner that is equitable in terms of racial, ethnic, gender, and geographical representation. We are aware that this is only a baby step towards addressing the disparity in scientific visibility, which we all know exists in our field and in many other fields. But we hope that our authors will enthusiastically take it up, and that we can make a bit of a difference here. So how does it work at Nature Reviews Bioengineering? As for the other Nature Reviews journals, most of our content is commissioned. That is not to say that we're not welcoming proposals, especially now at the beginning of our adventure, but most of our content is commissioned. So we meet in the editorial team and discuss the topics that we want to cover over the next 1 or 2 years. We try to identify the best authors for a given topic. We'll then invite the authors and ask them first for quick and brief outline, just really the main sections that they want to discuss, the key 20-30 references that they think are important, and some suggestions for display items. Based on this, we'll then invite the full review and provide feedback throughout the entire process. All our articles will go out to external peer review. And then based on the referee reports, you'll be asked to revise and then we'll finalize the manuscript together with you. So what makes a Nature Reviews Bioengineering review? Our reviews really shouldn't just be a laundry list of everything that's been done in the field, but it should be authoritative assessments of a field. So really the information should be synthesized and filtered to basically take stock of where we are to point to where we need to go next. What are the key papers everybody should know in the field or entering the field? What has worked? What hasn't worked? And importantly, we want the articles to be accessible to non-specialist readers, and we'll help you very much with this through our editing process. So our articles should be accessible by engineers, scientists, clinicians, readers interested in translation and technology transfer. We have a very strong focus on our artwork, so we want good illustrations and examples. We have a dedicated art editor who'll help you with this. And of course, it should be an objective and balanced discussion to really take on a stock of this specific area. And also, our review should really build connections and find new angles. I always say good review should always teach you something new, even if you've been in this field for a very long time. So really provide new insights and be inspirational. What are the key future milestones in the field? What are the major engineering challenges that will need to be addressed? So say if a young researcher reads this review, just thinking about their postdocs, that they get really good ideas for their research projects, fellowship proposals, and future collaborations. In addition to reviews, we will also publish comments, and here I've put out a few comments that I've handled in the past when I was at Nature Reviews Materials and at Nature Nanotechnology, just to give you a bit of a flavor of what we're aiming for. And our comment section should really be a platform for discussion. Everything the bioengineering community is concerned about, cares about, is discussing, should be reflected in our comments. And I particularly encourage young researchers and young scientists to submit some suggestions to us in terms of comments. We'll also try a new article type called Down to Business. And Down to Business articles are short, more common type articles. And the idea is that experts discuss how they took their research all the way from the initial idea and fundamental research to a product, including regulatory considerations, IP considerations, scale up and manufacturing considerations, and at what stage they started thinking about the spin out a startup company or industry collaborations. And our wish or our hope is that these articles can really outline honestly the scientific and technical challenges along the way so that others in our field can learn from it. So who are we? This is the editorial team. It's myself, Sadra Bakhshande, and Nesma El Sayed Ibrahim. We are all three full-time professional editors. We've all done our PhD and our postdoc, and but we've left academia now, and this is our main job. We're all based in Berlin, in Germany, and we also don't have an editorial board. So it's the three of us who are running the show. And what do we bring? I always say we bring you the Nature Reviews treatment because our only job is really to care about your paper. So we give you extensive editorial support throughout the process. We have the time to think, to discuss and work together with you to really make your manuscript into the best possible review in this area. I mentioned already that we have a really strong focus on artwork. So we have dedicated art editors who are incredibly talented graphic designers and who can work with you and with us together to create fantastic figures for your review article. And I wanted to give you an example here of a wonderful reviewer I handled at Nature Reviews Materials from Susan Thomas and on lymph node drug delivery, just to give you an idea of what we can do. So she wanted to have original schematics showing how you can target different compartments or cell types in the lymph node. And I remember I met her a few years back at the MRS fall meeting in Boston, and we discussed this. And she said she doesn't want to spend her students or herself spend too much time on Adobe Illustrator or something because they're really not good at this. So she decided to send us a hand-drawn sketch. And together with our art editor, this is then what we made with this. So a really new original schematic that you can then use for teaching, you can use to really better understand complex scientific problems. We also line edit, as we call it, every article, every manuscript before we publish it. And this is an example of a line edit I have done. That's Willem Mulder's review that I published in Nature Reviews Materials on regulating train immunity with nanomedicine. And I just wanted to show you this as an example. So we literally go through every article line by line and make sure it's precise, it's to the point, it's scientifically accurate. And after all, we want it to be a good story. We want people to read the article. So we'll spend a lot of time on the editing part of the article. And in the end, we always also may suggest a few additional article elements, for example, to include timelines to better illustrate the history or the parallel history, as in this case, the review by Yishidong and Bob on lipid nanoparticles for mRNA delivery. And also boxes, for example, to concisely explain important concepts in the manuscript where you don't want to spend an entire section on, and of course tables if you want to compare clinical trials or data of different studies. And finally, we'll publish it. Again, I'm showing you examples from Nature Reviews Materials because we haven't published anything yet, which is going to change very soon. So we publish it and we'll make sure the word is spread, we'll make sure people read it and that it really has an impact. And speaking of impact, I would now like to start the discussion with Bob and Annie about the real world impact of bioengineering. And I'm so happy that you both could join me here today. So Bob Langer, I don't really think he needs an introduction because he may be one of the most famous bioengineers. But Bob is a David H. Koch Institute professor at MIT, and he has published numerous papers and patents, many of which has really changed the way our field is heading to. And importantly, he's also co-founded many companies, including Moderna, who have been the ones who've developed one of the COVID-19 mRNA vaccines. So he knows a lot about fundamental bioengineering research and the translation and impact of bioengineering research. So thanks so much, Bob, for joining us for the discussion today. And Annie Moisan is the Hope Program Director at Wellcome Leap, where she leads a multidisciplinary team, really liaising between academic research groups, pharma, and startups, with the aim to translate bioengineered platforms for drug development and organ transplantation. Annie has worked in academia, in industry, and now at Wellcome Leap, so she knows a great deal about the different aspects of how bioengineering can make a real-world impact. Thanks so much, Annie, for joining us. And I would like now to start our panel discussion, asking you, Bob, what do you think were the greatest achievements in our field and sort of what impact these achievements have made?
Sure. Well, thank you so much. It's a pleasure to be speaking with you. Well, and actually, just so the audience knows, I didn't know I hadn't seen Christine's presentation in advance. So, but I did actually do some homework. I did see her questions in advance. So, but actually, I was going to highlight one area, which is actually the very first one she highlighted too. Partly it's because I'm highly prejudiced because I've been involved in this for about 50 years, but and then I'll also mention some others as well. So, and that is the COVID vaccines. But really, if you go back to that, I mean, this is actually when I first started my career, and it was kind of very basic research on how blood vessels worked. But we discovered a way with Judah Folkman, who was my postdoctoral advisor, of taking large molecules, including, this was the first time, to my knowledge, nucleic acids were ever put in tiny particles. So we came up with a way to take tiny particles, in this case polymers, and showed that you could deliver pretty much anything. And we even showed that you could deliver nucleic acids for up to 40 days. Now, I should point out a couple things. One, we actually did publish that in Nature. This a little while ago, it was 1976. But when I first started talking about this, it was widely disputed. A lot of very famous scientists told us that this was impossible. And the consequence of that for me is I got my first 9 grants rejected. I could not get a faculty job at a chemical engineering department, which is my discipline. I got rejected repeatedly. And I almost lost the job I got in the nutrition department because people didn't believe it was possible. But that being said, the evolution of it following that paper, several years later, a group led by Mark Ostro published a paper where they showed you could take lipids and do the same thing that we did with polymers. And then lots of other papers would continue, papers, where they showed you could take cationic lipids. This was out of California, and that would be very useful for nucleic acids. Papers that we were also involved in, where you showed that polyethylene glycol could be very important in preventing aggregation and macrophage uptake. Papers that were where you could even figure out the right ionizable lipids and so forth. So there was a lot of work over really almost 50-year period on this. And then, of course, several companies, CureVac, which was actually an early company, then BioNTech, and then the one that I, as Christine mentioned, helped start, Moderna, trying to apply these principles to now messenger RNA therapeutics. And of course, nobody could have anticipated this, but in 2000, of course, 2020, we have COVID, and these technologies made a huge impact on the world because they were clearly the first and best vaccines that you could use. And, billions of people have use them. So to me, that's been a great, like I say, I'm very prejudiced about it, but it's been a great example of what bioengineering can do. Just to mention a few others that I think have been very important, and some of these go back a long way, like artificial organs, starting with William Kolff's artificial kidney, but there are many others. Pacemakers, drug-eluting stents, defibrillators. The whole area of tissue engineering, again, I'm prejudiced because myself and Jay Vacanti helped start a lot of that. And that also, by the way, leads not only to new tissues and organs, but organs and tissues on a chip, which I think could be very useful for drug testing. Synthetic biology is another big area, protein engineering, directed evolution. Then I think there's areas that just understanding the body better, like understanding the vascular system, including work where it relates to tumors, understanding the GI tract better, including how it does relate to drug delivery, and many, many more. I'm sure I've left out a tremendous number, but it's exciting because I think it keeps going, and sometimes you find that things that have been done many years ago can have a big impact on what's going on today. Thank you.
Thanks so much, Bob. And I love to hear this story of when you say that at the beginning, when you started working on the lipid nanoparticles technology for delivery, the people said it won't be possible, and then you did not receive a grant and had troubles getting the faculty position. So how did you persist? How discouraging was this? How did you still manage to keep on going with the technology? Like what was the driving factor? How did you do this? I'm sure many of our young listeners would be very interested in this.
Yeah, well, I mean, first of all, I think you have to be very stubborn. Secondly, I was very lucky in the sense that my postdoctoral mentor, Judah Folkman, who really made the discovery about how blood vessels grow in the body, he faced the same thing. So I think having watched his example and having him as a role model was very helpful. But, it was very depressing. I think to me, I just believed in what I was doing. I could see these large molecules, in some cases we use dyes, come out with my own eyes. So I kept doing it. I kept writing grants. I'd learned things about how to write them, because I realized, like a lot of people felt when I first did this, that an engineer had no business doing biology things. I think it was, so people felt that that's why I didn't get a lot of those jobs and why I didn't get a lot of those grants. In fact, the biologists felt an engineer, this is, like I say, 1974, biologists would feel an engineer didn't know anything about biology. In fact, one of my grants said, well, Dr. Langer's an engineer, he shouldn't get this. He knows nothing about biology and even less about cancer. I thought I knew enough about math to know that wasn't possible, but it didn't really matter. And the engineers, they said, well, he shouldn't get a faculty position. He should be doing energy or oil or things like that, or mathematical modeling. And so that was discouraging. So I was lucky to get a job in a nutrition department. And I had good friends there, like Mike Marletta and Alex Klebanoff, and they also gave me a lot of advice. We were all growing together, but it wasn't easy. You just have to be very, very persistent.
I'm sure many bioengineers working in immunoengineering can sympathize with this, being told off that they shouldn't do this because they don't know enough about immunology. Annie, may I direct the question to you as well? What do you think were the great achievements in bioengineering thus far, and what was their impact?
Yeah, thanks, Kristen. So obviously we could go on for hours on the phenomenal portfolio of biomedical tools that bioengineering has created, how it has increased quality of life, especially since the Second World War. But I thought maybe I add to what Robert has nicely introduced, a bit of simply what I have experienced in my real world as a scientist, and as you mentioned, transitioning from academia to industry in the biopharmaceutical industry, and then to Wellcome Leap. So I trained as a biologist. I did not train as a bioengineer. And the toolkit, though, that bioengineering innovations has provided us since my early days in the lab some 20 years ago is, I think, a revolution in itself, like really. Genomic sequencing and editing, cellular reprogramming and programming, single cell and tissue omics. All these together allow us now to interrogate human biology like we never did before, and we can address mechanistic questions in powerful human-based systems. So in the same way as we can do it with, or we used to do it with, still do it with model organisms such as yeast, Drosophila, zebrafish, and mice, but now we can really combine stem cell, organoids, synthetic biology, biomaterials, 3D printing, microfluidics. You introduced all these concepts in the introduction, Kristen, and also Robert, but these all together make it possible. So we really study role and effect of specific genes and pathways in complex biological processes in a human relevant system, organ development, disease progression, endocrine responses as well. We can connect tissues now that can really crosstalk in vitro, and drug testing, these very complex responses to therapies that we can predict. Now we really have the hope we can see that in our in vitro system, and these new capabilities together with preclinical and clinical insights, so coming from the real world and those experiencing those problems. That altogether revealed how unique, I think, human biology is. We often see that as how conserved mechanisms are, and that is true, but subtle differences at the molecular cell levels can dramatically impact manifestation of a pathology or responses to therapies. And we learned it the hard way in the clinic and in pharma. So bioengineering has been determinant in making this realization, and I think it's also it is definitely part of the solution. And we are just at the beginning now of a paradigm shift in the way we use both animals and bioengineered human tissues to close this. You mentioned, Kristen, the so-called preclinical gap, like this infamous 90% of failure rate of drug development, which is a real world problem that bioengineers can definitely contribute to find a solution to. And then quickly, perhaps, if I can just add my perspective also on the fact that bioengineering is inherently applied. I do think that most transformative bioengineering advances also result from combination of both basic and applied sciences. And this is probably particularly true in bioengineering. So really fundamental life science discoveries together with engineered innovation, inventions that emerge at different times, converge really timely, and that is very powerful. And it's not, I think it's rarely pure serendipity, but definitely the hard work of scientists, engineers that came to think or talk or work together at some point.
Yeah, no, I absolutely agree. And you mentioned a variety of exciting techniques and approaches, including omics and manufacturing techniques. So would you say that a bioengineering lab, or in order to find bioengineering solutions, you will need a multidisciplinary team, including biologists, including engineers all together? Or would you say that bioengineering now has evolved towards its own sort of discipline? So would you call yourself actually a bioengineer, or would you say I'm an engineer working with biologists, or I'm a biologist working in an engineering lab?
Excellent question. So I do think you need a multidisciplinary team, but that doesn't mean you need all these people in one lab. And a bioengineer will probably find also an expertise. We see now immunoengineering as perhaps a new branch of bioengineering. And those immunoengineers I work with, they just like their knowledge of immunology is very impressive, and also the knowledge of bioengineering and the engineering principles. So the field is evolving as needs are emerging, and we are addressing the needs. And it's just beautiful how when we get started with a project, with a question, we need always to embark more and more and more people. And I think this open-mindedness makes this team powerful, and that's the way to do it. So I don't know how a bioengineer would just kind of introduce themselves and qualify themselves, but I do see, sure, bioengineers also being or deciding to be a bit more of a biologist or material scientist, for example, but there's certainly room and need for everyone in there.
Bob, would you call yourself a bioengineer?
Well, people have called me a lot of things, but that is one of them that they've called me. I mean, I'm technically a chemical engineer, but actually I have appointments, I think, in 6 different places at MIT: chemical engineering, bioengineering, mechanical engineering, the Cancer Institute, the Media Lab, and the Institute for Medicine and Engineering Sciences. So, and then I've been on the boards of both the Whitehead Institute, which is a biology institute, and the McGovern Institute, which is a neuroscience institute. So people end up calling me a lot of things. I've actually also been called a biochemist, which I'm hardly qualified to be. I'm not sure I'm qualified to be any of those things, but people have called me a lot of different things. I mean, and those are just the technical names.
So you both mentioned many of the exciting bioengineering research that has been going on, and however, many of those have not made it out of the lab yet, and we often face or face in past the problem of getting bioengineered solutions translated, getting regulatory approval, getting it into a product. So what would you say, Bob, are the key hurdles to the translation of bioengineering research? You mentioned your story with lipid nanoparticles, of course, and this was a very long process. And ultimately, I don't know if you would agree, a pandemic made it possible to really get it into billions of arms, right? So what do you think are the key hurdles?
Well, sadly, I guess I'd have to say the number one hurdle is money, funding. I mean, and part of that is just, if you try to start something like Facebook, it doesn't cost that much money, right? But if you try to, what is it? I think Tufts, for example, estimates that if you try to create a new drug, it costs between $2 and $3 billion, and that's if you're successful. So, I mean, funding is just a giant issue, and to me, raising that funding is always a challenge. I mean, you can get things started in the academic world, and I think we do. And I agree with what Annie said. I think you want to do kind of basic research aimed at trying to understand things and solve problems. But to translate that, there are a lot of different issues, funding being the first one. Others, I think, are, and some of these relate to funding, having good investment partners, people who can, who have funding, but also can give you good advice when you do this. In particular, having good chief executive officers, really good business management team. I think that ends up being important. Sometimes you can try strategies, which we've done in cases this now relates to the FDA. The bar is very high to try to create, things that will get approved. It's costly and takes a lot of time. And sometimes even after people have spent a lot of time, they still get rejected. And so I think having good regulatory advice, good people who can help on that is important. I think there are actually many things that there are many barriers that occur, and so it's challenging. But a lot of it does come down to funding and leadership and really good advice and highly committed people.
You mentioned, you both mentioned actually the importance, of course, of basic research. Would you say, Bob, that it is very difficult to get funded with a bioengineering project without really having a specific application in mind?
Yeah, well, that's a really good question in a number of different ways. You know, a lot depends where you get the funding from. I'd say, you know, so foundations, like if I look at our lab, you know, we've gotten money from the government, like NIH, we've gotten money from the Gates Foundation, we've gotten money from specific foundations like Juvenile Diabetes Foundation and Helmsley Foundation, and also from companies like Roche and Novo Nordisk and Alnylam and so forth, which is on there. So everybody uses different criteria. And I would say for the government grants, I think you can get funding for basic research, but it better be very good. And you probably, in my opinion, should have done most of it already. I mean, in other words, they really want you. I still remember one of my first grants. My first grants all got rejected, but when I finally got one, I still remember the review came back. Well, maybe we could give Dr. Langer his 12 papers that he just published on this, showing that he could do all this. You know, makes us think that maybe it's possible. But so you have to do a lot. So when I mentioned some of the other things, if you're doing any of those foundations, they all have goals. So there, I think you do want to have an application. Though I'd say, you know, we've been able to do pretty basic engineering research with some of the Gates Foundation money, but we've always had a novel application that could help people, you know, maybe vaccinate them better, give better nutrition. Companies, I think a lot of it, to me, has always been for us having patents that they felt would be useful. So we've been able to have patents, and that's made a big difference. And so much depends on. I always use a baseball analogy. You know, or football. You know, we're playing in somebody else's ballpark, and they have different rules. So I think it really depends where you have to play by their rules, and so I think that makes a big difference of how you set things up.
So Annie, what do you think, from your experience, what are the key hurdles through the translation of bioengineering research?
Yeah, there are many stakeholders that need to be convinced, right, for a successful transition of an invention into real development. So and funding obviously is that it starts with funding most of the time. So let's assume that we got the funding right, and that's what HOPE. Programs at Wellcome Leap, I think we have mechanisms that aim to facilitate this transition, and I can explain like two main ways that we do that. I think that can be insightful for the audience here. So the two ways that we construct and orchestrate our program focused on this transition and being convincing. So first of all, we defined the real world problem that we want to solve, right, as a program. And we use analytical frameworks to articulate common goals. So the goals that we have are ambitious, but they are focused, testable, and measurable. And this is important because we can also measure, you know, how we make progress and how we always like target the common goals that we have. Second, we work with system demonstrations. And system demonstrations are experimental challenges against the program goals. So they provide evidence at convincing scale. So these demonstrations point towards like new possibilities of what was previously thought impossible. Okay, so they are not finalities, they are just key advances that can change minds. And that is key here, because by allowing such, we can then unlock the investments to get the discoveries and capabilities out of the lab. And if I just have time maybe to give an example, I think this is a bit abstract. So problem that we have, people die every day awaiting for organ transplant. So this is because we still haven't found sufficient supply of functional organs, and we cannot adequately prevent rejection, right? So at HOPE, we ask, what if we could double the 5-year survival rate of patients awaiting for a kidney transplant by bioengineering functional universal renal units? That's our goal, that's our question. So when we do the homework, we found that it's clinically estimated that providing a filtration rate equivalent to 10% of a healthy kidney is sufficient for patients with advanced CKD, chronic kidney diseases, to be taken out of dialysis. So that is our measurable goal that we can do, with a bioengineered living kidney. So as we then grow organoids-based human kidney, we will measure the capacity of these artificial but living tissues to contribute to renal filtration. We do that in mouse models of renal insufficiency. We can't do that in human, but we keep working on that until we've achieved, this bar that we said that we understand has a real impact or should have a real impact in the clinic. And then in parallel to that, we build the biomanufacturing capabilities so that we can scale up then in size and function. So we estimate that we need 50 times more, from mouse, scaling from mouse to human. So with these demonstration and capabilities in parallel, we make then we can claim we make the key advances to that are necessary to produce a universal living functional kidney at human scale, right? So we work on the early steps indicating clinical feasibility, and we think that with that we will convince the next stakeholders. So it's really in the approach, in the thinking, in the preparation, in the mechanism. So we act early, but we really want to convince and unlock what comes next to facilitate this transition out of the lab, if that makes sense.
Yeah, no, very, very interesting. And thanks for the really good example. So as part of Wellcome Leap, you work with academic researchers, but also with researchers in industry. And obviously for academic researchers, publishing plays a huge role, being it obviously to disseminate your results so that others can read those and learn from those, but also it's a huge, it's often related to their careers and to funding and so on. So I was wondering how much you think the publishing landscape enables or hinders perhaps sometimes translation of bioengineering research.
It's a very good question, because indeed the publishing and funding are, most of the time right now indissociable. And that means, or consequently, I think that puts, upfront the individual's publication record. And there's also, like, coming along with that is some sort of popular storyline of a lone hero scientist who pursued new discovery until a breakthrough, and it's a lot like on one person taking the credit. And that together makes it quite hard actually to work in what we call the use-inspired space, right? This translational space, because working there requires, a network of diverse contributors, synchronized, multidisciplinary network. Momentum is really key. And it's challenging for an outcome-oriented team to keep this momentum and publish regularly at the same time, knowing what it means, right, to publish, and the timelines and the rebuttal, and also the time we spend, the tensions, debating over authorship, number of papers we need. So sometimes it's actually a blocker. We decide not to, depend when funding is secure, often we decide not to publish. That's also what happens in pharma, even if there's no intellectual property or proprietary data. It's just, we need to move on with what we have in mind, which is demonstrating an impact and really contributing on a project, a portfolio, a program. So publication then becomes a bit of just like a nice to have and not a must have. But I do think it's good to think about how can we perhaps in publications value a bit more the team's contribution. We often discuss how we should work more efficiently as a team to demonstrate translation, to address the key questions. Can we also in that dialogue include like publishing and dialogue with publishers? So what would be the mechanism so that also there is value in work that is so important to make the final step, to demonstrate robustness and reproducibility, automation, all these components that are required for adoption of a new method. And adoption means impact. If there's no end user, there's no impact in the end. And we often very focused on the early stages, that discovery, and then we dilute, or we don't value as much what needs to be done next. It's perhaps perceived as not novel, although I'd say it's equally important. And without all that work, and not everyone can do that work. It's really, you have to be tenacious and resilient to be able to do that and take it to the finish line. So I don't know if that answer your question, but I think publishing is can be indeed not so well aligned with that ultimate goal.
Yeah, absolutely. Bob, what's your opinion on the publishing landscape in our field and its role in translation or impact?
Yeah. Well, I might be in the minority, but I'm personally a big believer in publishing for a lot of reasons, even for companies. But I realize, and I agree with what Annie just said, sometimes if you're at a company in particular, it's difficult because you're trying to get products out the door and you're trying to survive and grow. But that being said, to me, I've always felt the benefits are significant. Certainly if you're in academics, the benefits are obvious because, it helps on grant support. And sometimes people think you can't get a patents when you do that. But I mean, I've always used like early drafts of papers as the basis of patents and try to have our licensing office tell the lawyers to get to work right away. But I also feel that for companies, it's valuable too. I mean, I'll pick a negative example and then some positive examples. I think a negative example, unfortunately, was Theranos. They didn't publish, and a lot of people, and a lot of people ultimately felt that hurt their credibility, which I'm sure it did. And so people started criticizing companies when they didn't publish. I know even with Moderna, there was criticism, even though I think they didn't realize that we did publish some things, but and Moderna's published plenty of things now. But positive examples, I remember when I was an advisor to Genentech, and I was an advisor for many, many years, they published a lot. And I feel it actually had the benefit of when they published something, their stock went up, investors thought it was very useful. And, and for small companies, when you publish things, I think that can also attract both investors and big companies to say, well, gee, maybe I want to learn more about this and maybe fund your smaller company. So I think there's definitely benefits about it, but you have to weigh those benefits as against the ones that Annie mentioned, which could be negatives. But I personally almost always recommend the companies I'm involved in to publish, not to not patent. I'm a big believer in patents too, but to try to do both.
I'm glad to hear that.
I fully agree with that as well. Definitely the message is also like positive in publishing. There's clearly gains in publishing work from industry. That definitely, yeah.
Yeah, no, and I think with the launch of our journal, but in general also, I think there is much more appreciation now for the application, right? And as you mentioned, for the impact, for the more user-centered or application-centered approach. And I feel like that has changed in recent years. And we definitely at our journal have a huge focus on this. And we also have authors from industry, for example, writing reviews, because I think it's important to get this perspective and to know what's actually in the pipeline, right? Already in the translational pipeline. I want to switch gears a little bit now and talk a bit about the future of our field, and be interested, Annie, in where is our field going in the near and long term? What do you think will make the biggest impact? What will be what will be the key amazing new bioengineering things we'll see over the next years?
Well, the biggest impact, I don't know, but there are so many areas where I'm convinced that bioengineering will make an impact. So bioengineering right now is creating living solutions. And the scope of application is immense, and living therapies, sensors, vectors, and together with synthetic biology, artificial intelligence, our ability now to program biology, I think that is going to change the pace and economics of bringing new medicines to to patients. I'm excited about keep increasing the clinical relevance of in vitro human models. One way I'm thinking is integrating population diversity on chip. This has been proposed by many. I'm not the first one to propose that, but I think, to make that possible, we still need leap advances in cell and tissue sourcing, biomanufacturing, and chip optimization and production. But we could have, we could build like versatile, miniaturized non-clinical platforms at high throughput and high predictive power. And this is something that we cannot achieve with animal models. It's just not possible. Then you can really propel our understanding of inter-individual responses, discover better biomarkers, make personalized medicines possible, but also at perhaps more favorable cost. I like what you said earlier. So can we also have solutions that allow lowering the cost of all these new inventions and innovation? And I'm also looking forward to see how coupling some more bioengineering with real world evidence. So that could be an accelerant, thinking here of simultaneous advances in wearable devices and biosensors together with AI, machine learning, that can really capture the real world outcomes, of a therapies, for example, at scale and at speed. So to really then provide direct insights, and that could feed engineering principles of what we're doing. So of new and existing biomedical solutions, and eventually address a much broader reach of end users and patients. So I do think that in the next 5 years with bioengineering, we will rediscover many aspects of human biology. And in 5-10 years, I think we can also invent new biological systems that will be fit to solve existing and future problems. So the COVID pandemic taught us the importance of preparedness, speed and agility of actions. And I think bioengineering will be there and play an important role there in many, many ways.
You mentioned a bit of personalized medicine and the opportunities that there are to program biology and to use the chips and the devices we create to test human disease or investigate human disease and also for drug development. You also mentioned costs. And I mean, this is something that comes up a lot, right? These are all very, very expensive approaches, at least at the moment, speaking of organoids or organs on chips or tissues on chips and all these kind of creations. Is it something that we need to consider as bioengineers early on in our research to ensure we're not creating something that ultimately will only be used in 3 labs in the world because nobody else can even afford experimenting with it?
Yeah, very good question. I think you should definitely never aim to produce like a one lab invention. It has to be to go out there. So yeah, of course, it's the number of end user and the broader adoption will lower the cost. And as we work together, this whole community, also anyone working on cell therapy, tissue therapy, all the manufacturing aspect of tissue engineering, so lowering the cost, increasing robustness and reproducibility, demonstrating that if I can grow an organoid manually, then I can grow like 10,000 organoids with an automated platform that reduce the cost. And can we think, there are so many aspects of it I can just think of. We use like growth factors, these are very costly. Can we then have a focused effort on how do we lower the cost by moving away from growth factors to like small molecule based differentiation? There are tons of aspects. So we need money to get there. We need time. We need a lot of effort. So it's not for tomorrow morning that this is a low cost exercise, right? To move away from animal studies to human based systems. I think pharma understood that, and that's a limitation. We had to manage expectations, right? But it will be possible. We have to make it possible. So the technology will come along, and that's where we will have breakthrough, I think, to make this happen.
Thanks, Annie. What are your thoughts, Bob, on the costs of bioengineering research and the way we develop these systems?
Well, the costs are certainly high, but there's probably 2 points I want to make in terms of what Annie and you just asked. One, I mean, well, certainly what Annie said is right, that it's not cheap to do organs on a chip or tissues on a chip, but it's actually a lot cheaper to do it that way than it is to have, the same number of animals and the same number of people. So I still think that that's highly worthwhile if you can do it, and then you can do high throughput things. So I think that's really important. Also, to what you said on the question to her originally was, if only 3 labs can do it, is that still worthwhile? Well, certainly I would agree that if that's what it's going to be 100 years from now, then of course you shouldn't. But I don't think, if you look at history, that's the case, right? If you do something that's initially expensive, let's say like gene sequencing, right? When people first started to do it, it was unbelievably expensive. Now you have companies like Illumina, 10x, and others, and it's tremendously cheaper. And I think and there's other technologies that I could speak to that are the same thing. So I think getting a proof of principle, particularly when people don't think it's possible to get that proof of principle, I think that's a good thing, even if only a couple places can do it, because I'm of the belief that once something's demonstrated and people's beliefs are changed, that that people will figure out how to do things cheaper. Just as a personal example, we worked on things for many, many years in our laboratory. As I mentioned drug delivery and others, but Bill Gates and his team came to see me 10 years ago, and they said, well, gee, we think that's really important. We'd like to give you some funding to adapt these technologies that you've developed for the developing world. And I thought, great. I mean, that would be wonderful. And we've done that. We actually have nine clinical trials, maybe 10 clinical trials that we've started like in only 10 years from developing new technologies. By the way, we published a lot of this stuff in Science and Nature journals too. And we've started companies that the gate like Lindra, which is developing pills that you can swallow that can last a week or a month or the entire course of treatment. Some are aimed at what should I say, rich world, as they use the word, rich world applications, like treating schizophrenia and things like that. But we're also working on new treatments for malaria, new treatments for birth control, that the Gates Foundation even funds the companies to do. And so I think that once you demonstrate something can be done that people think is very unusual, then I think there are ways to figure out ways to reduce the cost and make it more and more available. So so that would be my 2 cents on that.
And of course, I'm also curious now to hear your thoughts about the near and long-term future of our field and where you think we're going.
Well, I think the field has, of course, again, I'm highly prejudiced, but I think it's got enormous growth potential. I mean, all the areas that I mentioned before in your very first question, I think, I don't want to say we've scratched the surface, but we can do so much more on nanoparticles that you could target to specific cells, that you could deliver through different barriers like the blood-brain barrier. And I think, as Annie mentioned, like there's so much you could do on artificial organs and tissue engineering and synthetic biology and things like that. So, but let me even mention other areas that I think can be important in the future. Like I think applying engineering to neuroscience, like we're working with Li Wei Sai at MIT and Alice Stanton, my postdoc, on creating brains on a chip. There's people like Arup Chakrabarty and others at MIT that are doing immunology and trying to better model the immune system. And then I think there's work, again, I've seen at MIT, and actually it started in Germany, a lot of it, where you can apply engineering and physics to cell biology, like with condensates and things like that. And then there's, as Annie mentioned, and you may have mentioned, artificial intelligence, digital medicine. I mean, I think there's just so many areas, wearable devices that engineers have played a role and will continue to play a role. The last point I want to make, though, on this is it does go back to basic research. A lot of times I think doing curiosity-based research on things that don't even have a name, but I mean, and I'll pick an example in a second, but that don't have a name, but really could yield potential benefits by understanding things. I think, just because something has a name like tissue engineering or drug delivery doesn't mean that that's what you should necessarily work on. I mean, just trying to make basic discoveries, I think, is important. And the example that I was going to pick is CRISPR, right? If you asked almost anybody 10 years ago what CRISPR was, well, nobody would have known, because most of the most important or the key papers weren't written. So I think people following their dreams and doing, really good basic research in engineering, maybe at the crossroads of engineering and biology, or creating new tools, I mean, those could have a huge impact.
I'm glad that you said this now, because my next question to you would actually be, what would be your advice for young bioengineers in terms of, publishing, funding, translation, all the things we mentioned? What would be your advice for them? What to focus on, or how to go about it, given your experience, longstanding experience in this field?
Yeah, well, when I get asked to give like commencement speeches and stuff, usually what I do is I go back and I always try to say to people, and I give that example of myself, the getting rejected all the time, but or at least for a long, long time. And I really think it's important to not do incremental research, to do, a lot of times when people start out, they say, well, geez, I need to get a publication. I need to, get a grant. But I really feel it's worthwhile to try to dream big dreams that can change the world. They could be in anything. And try to make discoveries that will have that kind of impact. I think that there's two things I'd say. If you do that, you'll probably get rejected a lot. And a lot of people, even if you do succeed, will criticize you. But I wouldn't give up. I certainly would not give up easily. And so that's one of the things that I would do. I also, and that's probably the biggest message that I would give. But I also would say a few other things. I do think it's important, and you raised some of these points before, to try to get significant funding. And I think when you do that, look for funding in unusual places, not just the standard and in unusual ways. Like it could be patents, could be foreign governments, could be companies, could be foundations like Wellcome Leap or others, and I think that you want to really, make a priority on getting funding. I also feel that another point that came up about what I call convergence, and Phil Sharp and I have written articles on this, getting multidisciplinary, this is for bioengineering specifically, getting multidisciplinary teams, whether they be at the same institute or different institutes, to work together to solve problems. So I think those are some of the things that I would recommend. I also think getting advice early on, having role models, having people in your department or previous mentors that you can get advice from. I think it maybe even review some of your grants, I think is important.
Thanks so much, Bob. What would be your advice, Annie, for young bioengineers?
Yeah, I really like to be asked this question, and here especially because it's about advice for young bioengineers. And not being a bioengineer myself, but I did have the privilege to evolve with the bioengineering community, and it's incredible what I've learned, from such a diverse and vibrant group of engineers, biologists, physicists, computer scientists, entrepreneurs all together. So what I sincerely want to say is, everything is possible. So ask the big questions in the area that excites you most. Then I'd say focus on what is possible, so what you can do there. You will realize that there are limits to what one can do alone. So seek for your allies, reach out. And don't think about the obstacles. There will be obstacles, but you also will not be alone in tackling these challenges. Be curious and don't be shy. I say that because, and I want to say it's fine not to be the expert in every conversation. So if you want to navigate a multidisciplinary team and topic, at some point you have your own limitations. So you know what you contribute and you know your strength, and then you listen and you communicate. And this is extremely rewarding in my opinion. And it's you have to just accept not to be the expert, and that's fine. And then on the publishing aspect, if I have an advice, I think it's wise to reflect on a publishing strategy, starting a project and starting in science, and I'd say discuss with a scientific or career mentor. Try to understand what you want to achieve by getting your work published. I think we do have different goals in publishing as well. So what do you want to reach, or who do you want to reach? What is the take-home message? And perhaps you wish for your work to be continued by others, augmented. You want to inspire other researchers and contribute to building a community of people with similar aspirations. And I think that, in publication, our collaborative mindset is also manifested. Also, the way we handle response to publications and address inclusivity and in the work in our reach. So publishing is definitely key. And then I think we all know that the technical skill set is something, and it's important to be an expert, but also in addition to this skill set, what else do you want? What else do you think you need? Maybe some entrepreneurial, legal knowledge, or knowledge in another field, basic knowledge or applied, IP protection, for example, communication. I do think communication is always key, influential skill, depending on how applied you want to do the work. I also found quite rewarding to get closer to quite inspiring, get closer to clinicians as well, if thinking for applications in the medical or the clinic. Seek for collaboration with those who are experiencing the problem. Industry, it's definitely possible to do a postdoc in industry, to exchange with industry people, and work together, publish together. So I think there are so many possibilities. It's not so. Go outside of your comfort zone and your lab. I think that's the advice.
It sounds wonderful, Annie. And before we get to the questions from the audience, and I've seen we've got quite a few, selfishly, I'm going to ask you now what you would wish from our journal to cover and publish, or anything. What would you wish us to do? What would help the bioengineering community, and in particular, like the areas you're working in?
Who did you want to go first?
Bob, you go first.
So, okay, okay. So anyhow, I actually made a list of this before. Again, I saw your thing. So I looked at thought about other journals that I saw things I like. So I was going to mention, I like that bioentrepreneur part of Nature Biotech, but your down to business idea sounded good. I like that. But maybe you could also have bioengineer profiles, like or interviews. That could be good. And I don't know that they just need to be on senior people. They could be on other people. In fact, one of the things I've also liked, and now this is from Science, is they have this like careers thing, at the end, where they talked about different people. I mean, I've been in both of those, so I thought they both did a very good job. But it doesn't have to, again, be senior people, like old people like me. It could be younger people, or people, in different parts of the world, like you were saying, and so forth. And I also think interviews, could be useful. And I think also you made the point, which I agree with, of wanting to have people in low resource countries and have equity. So I think that those could be people who could be interviewed, or all those things that I just mentioned. But I also think, and maybe you're doing this, is to solicit papers from them. In other words, when you're talking, like I've had people from Africa really want to try to get more involved in bioentrepreneurism, and so I've given lectures on Zoom about trying to do that. But I think that soliciting whether it's reviews or interviews, trying to get people, minorities, people from Africa or other places involved, I think would be great. And there are all kinds of role models too that would be helpful for lots of people to see, because there are plenty of people who have been successful from all walks of life.
That's great. These are great suggestions, Bob. And indeed, it's one of our aims to commission articles as broadly and as diverse as possible, and to really ensure that we're not highlighting the same few people, right? But to really do the broad nature of our field really justice. And I really like the suggestion of bioengineering profiles. That's a really good one. Annie, what would be your wish list for us?
Yes, I'm convinced that advances, trends in bioengineering will be very well covered. So what you introduced earlier, I think this is spot on. And so I am delighted to hear also that we think alike in being inclusive of different or like a broad geographical reach. I think to me that is very key to, again, to be impactful as a field in bioengineering, how to reach more globally. And then we need them to participate to the discussion and understand the real world, is very specific of where you live and what you experience. So we need to hear more about that. So that's what I wish to see in terms of authors and co-authors from various sectors and types of organizations and geographical location affiliations, a balanced representation, of technology developers and users, regulators, as you mentioned, is the scope anyways, public and private institutions in different countries. And I think overall this will be a great opportunity to bridge academia and industry, for instance, or low income with high income countries, raise the awareness of the real world problems that need to be solved and that we can solve with bioengineering. So some sort of uniting platform to remove some of the cultural differences and prejudices. I think the journal could do that, and that would be great. And I think this is also a great question to ask to the audience. I'd like to hear them about what they wish to see and to read about in this journal.
Thanks so much, both. And this is definitely something we're striving to do. It's challenging, but it's definitely our aim. And absolutely, if there are any wishes or suggestions from the audience, would love to hear them, of course. And I now want to take a few questions from the audience and considerate of time. So I would hand over now to Sadra, who is an associate editor in the Nature Reviews Bioengineering team. And Sadra, I think we have some questions from the audience, right?
We do. Well, I think I'll first, though, take advantage of being a co-host here and just ask mine. I guess that's the usual way to do. I don't know whether it's correct or not, but I'll do it anyway. Well, I'll start with Annie. I'll try to give the questions a bit more of a personal spin, maybe, just to see, to cover the breadth of bioengineering, but also non-bioengineering aspects. And I would like to start with Annie, maybe. Considering that you have experience in an academic environment, also in a regulatory agency, and as well as a big pharma company. In terms of your working experience, and here I'm talking about work-life balance, how much stressful what were each of these positions and so on. What's the biggest difference, and that you would like to somehow make the audience aware of?
Wow, that's a great question, a bit unexpected question. There are clearly differences, and it's what I appreciated transitioning from academia, and I love working in academia. I think it's really rewarding, and the freedom. Yeah, we often want to stay in academia for the freedom. What I experienced in the industry was the acknowledgement of hard work being done on a very regular, weekly basis, and recognition of the work, of the contribution. And that relieves a lot of the pressure. So being appreciated not only for, the final publication, but the daily, the regular efforts. And also the the teamwork, which is necessary, and I think academia also progressed in the field of bioengineering is a good example of how this I think is an inclusive field, I think, in itself, which is not the case for all field of research. Personally, I really that's meant to be my ecosystem, is really when I can work with a team, when I can collaborate, when we have a common goal. And it's really in the benefit of everyone to work together, because we have to deliver. There's an understood, a common agreement on a deliverable, and that helps. And I think that is transferable to any setting also. If we have a good understanding, a clarity on the goal we want to achieve and what we need to get there, then we work as a team toward the goal, and that's quite rewarding. And for me, involving the regulators, I think that's a great experience as well. And from an academic perspective, the policymakers and regulators are important. They are there down the road. You can't ignore them, especially working in translational science, and they see a lot and they know a lot. Right, they're full of insights that are not in the public domain. So I think this is also a learning I can share here. Involve, like the rule in pharma about regulators, is involve them early. So you can only benefit from this communication with whoever the stakeholder is. I think what we learn in pharma is communication and messaging. Really, who is the audience? Who do I need to convince? What is the message here? And we work on that. So that's a great experience. I can really recommend trying. And it's not true that if you go in industry, you're stuck there, or can never go back to academia. And anyways, that could be a good place to be stuck, to be honest. There's no problem in that. But no, I do think, especially nowadays, you see this flow, this movement of scientists from academia going to industry, from industry. Perhaps we need more of industry people also going back to academia, or at least as mentor. That is very important, because we do in pharma needs scientists, a constant flow and fresh blood and fresh ideas. That's much needed, and I think we can foster that a bit more.
Very interesting. Thanks a lot. And actually, I would like to follow up the point that you made about the interaction that we should basically have, like people should have with regulatory agency and so on, and ask a question to Bob about your experience. Like, what's the difference in your experience, your interaction with regulatory agency, and in general in the whole translational process 20, 30 years ago compared to now? Look, are these agencies, is there anything that you would like to highlight in the, even the changes that has occurred on the other side, I would say?
Sure. Well, I mean, a couple of things. So I think from 1994 to 2002, I was actually on the FDA Science Board, and I was chair of it for 4 years. That's their highest advisory board. I would say, both from that experience and just from other things I've done as like helping get companies started and being on companies' boards, that it's really good to meet with the FDA earlier rather than later. I think that they can be very helpful to you. So I wouldn't make assumptions. I think that they won't tell you what to do. Like you can't just go and say, well, how do I get this approved? But you can go with them with a plan about what you're going to do for a clinical trial, or what you're going to do for phase 1, phase 2, or phase 3. And I think, and show them that, and have a meeting with them, and they will get a group of people and to do that. So I think it's really useful to do it earlier rather than later. I would say, in terms of the FDA over time, I think obviously technology's changed, but the FDA is really there to make sure, I think, things are safe. Various things in time, incidents in time, which people will be aware of, I'll give an example or two in a second, have made, I think, an influence on how FDA regulates things. But still, they're there as a watchdog to try to make sure that things are good. The examples I was going to say, if I were to pick two things, I think when the AIDS crisis came about, the FDA did lower the bar in terms appropriately, I think, in terms of allowing drugs to get approved, because if people were dying, you really want to think about that. On the other hand, when there was a Vioxx situation, must have been about 2005, where actually a very small percentage of people died from that drug. But still, I think what happens is when those things happen, Congress and lawyers, there's all kinds of lawsuits, and I think that made them more conservative again. So different incidents can have effects over time, rather than maybe just time itself. And politics can sometimes also play a role. I've been a big believer, and that was the goal of the Science Board, to make what the FDA does really science-based. Yet that might seem straightforward, but it's not always that way. As you may know, sometimes political things or religion can play a role. So I think that, and a lot will depend on the leadership and things like that.
Very interesting. Actually, since you mentioned here we have a matter of trust, actually. So it really depends on how much people also accept what is being presented by bioengineers. We have a question from Felipe, who's saying, well, beautiful conversation. Thanks for organizing this. Some of the technology that has been discussed is really advanced for late women and late men. Trust is a huge roadblock to effective interventions. So COVID vaccine is an example. How can we overcome, as scientists, mistrust in bioengineering tech?
Is that, do you want me to try to answer? Do you
Yeah,
want
maybe.
Annie?
Yeah, you could.
Or,
for example.
Yeah,
Annie,
how can do you want to go first, or do you want me to go?
You can go first.
Okay. So I mean, it's a really good question. I think the media can play a tremendous role. And, my feeling is talking to the media is for scientists is important, because they can play a role. And unfortunately, I think the media, certainly in the US, during the COVID crisis, there was something called National Bureau of Economic Research, and they made the statement or analysis that 91% of the articles that came out in the United States were negative with respect to COVID vaccines or COVID things like that, and that there were more articles on Donald Trump taking some drug than on all of the positive science going on. And I think, my personal experience was that there were a number of journalists that really, and even at good, what would you think would be good newspapers, who really I thought were very disappointing to me, and trying to be sensationalistic at the expense of, really giving accurate, information and so forth. So I think I would say talking to the media and trying to get them to do things right. And I also think this is, again, a reason for publishing. I think when you do publish in science and nature journals and places like that, those journals do get, do help a lot, because I think it gets credibility. Obviously, things like Theranos and so forth, places that don't publish, I think that also can hurt trust, because again, the news media can sensationalize those things, and have. I'm not trying to say they're wrong about that in that case, but or other cases. But I think that publishing, again, in scientific journals is very important in terms of helping trust.
Thanks a lot.
Trust is something that is built, that takes time, that relies on relationship. Trust is like real trust is mutual. So it's a challenge that we have as scientists to communicate and be trusted. It's we have a critical mindset and analytical, mindset. Fact-based works well for us, but that's not how we convince others. That's not how we build a trust, and we have to take a distance and listen. It really takes time. So I think in this, what we've seen in the COVID pandemic, for me, it worked at the more like the personal level, convincing people, explaining vaccination and why. And it worked because trust was there, or people came and said, I trust you, so what do you think? So big question for me, I kept asking myself, how do we then build this trust collectively with who do we need to involve and what are the mechanisms? So it was a lot of listening and trying to understand. And I realized, as scientists, we are comfortable in gray zones, like a cloud is not something that scares us. We can see and connect the dots and see clarity as there are more and more evidence, and then that can change from one day to another. But that's not the case from everyone. It's much easier to see black or white, and that's why conspiracy are giving people like some trust or confidence, something that to cling to. So we scientists can play a role there in rethinking how we communicate with the public, how we listen. What are the platforms for listening, for understanding their message? What are the fears and concerns and why? And always like re-explaining the same thing in the same way cannot really help. So it's not the solutions here. It's really a big challenge, but I think that's important to understand where we stand and the way to go.
Absolutely. Thanks a lot. I mean, we have only 5 minutes now, and we have a question for maybe Christine. Do you want to answer this one? I'll just jump directly to the last one. That most articles are commissioned by editors. This will favor big, famous labs. So while small labs who do good work have very little chance. How do you achieve inclusivity?
I think that's a fantastic question. And that's one of the, I want to say, biggest advantages of being a reviews journal is that we commission. So we can be very strategic and inclusive in our commissioning. And at the end of the day, we meet in our commissioning meetings and we mainly care about topics. First of all, we'll discuss the topics we want to highlight, the topics we want to cover, of course, based on trends in the primary literature, but also trends in translation, really, and also what's important now and in the near-term future. And based on this, our aim is really to ensure that we do the global nature and the diversity of our field justice. We'll try our best, and I've mentioned it at the beginning, I think briefly, but we always welcome proposals. So if you have a very good idea for a review idea, please feel free to reach out to us. We're happy to look at proposals, and we actually got quite a few very good ones, Sadra, right, last year. So we're very happy and really cool suggestions that we haven't thought about it in that angle, for example. And very often these come from younger researchers, right? Because they see a field sometimes differently than people who have been in the field for a very long time, and they provide exactly this new angle and food for thought that we are looking for in our reviews. So I hope that answers the question.
Awesome. Thanks a lot. So can I ask another question? We have 3 minutes.
So one quick question, and then I think we're unfortunately running out of time.
All right. So I'll just ask one quick question direct to Bob. Okay. So great discussion, Professor Langer. Why cancer immunodrugs are so costly per dose?
Why they're so costly? Well, I'm not sure I'm the best person to answer it, but a lot of times drugs are costly because because of the amount of research and money it's taken to get them to that point. So I think that what people, some of it goes back to what I said before. It may cost X to produce a drug, but it may cost 100X or 1,000X to do the clinical trials and all the work to get that drug in the first place. So from a practical standpoint, a lot of this goes back to what I said earlier. It's so expensive to get any drug out. So if investors are not investors, regardless of what sometimes the media says about the government funding a lot of things like Moderna, I mean, really, those companies would not exist without investors. So if investors are not able to, in some cases, investors have a choice, right? They could invest in Facebook or Twitter, or they can invest in Moderna or BioNTech. So and believe me, it doesn't cost much to write a Twitter or to do a Facebook. But the problem is, if investors, when they have a choice, they couldn't make money on it, well, then everybody's going to invest in Facebook. There will never be a Moderna or BioNTech. So I think the cost goes in because the clinical trials cost so much and the failures are so high. The rate of drugs failing, I think, and actually, Christina or Annie or others may know more than me, but I think it's something like 95, 96% of the drugs that even start clinical trials even ever get approved. So I think it's all of that makes drug pricing high.
Agree.
All right. Thanks so much for the great discussion. Thanks so much, Bob and Annie, for taking the time to share your thoughts and your vision for the field and your story. I think it was incredibly interesting, and I'm very excited to hear all the research that's going on and that's coming our ways, and how close we are really to translation and how much impact our field has made. And you both mentioned the importance of communication in our field in various areas, and that's where we, as a new journal, hope to play a role, and in communicating our research and giving diverse voices a platform for discussing all these aspects really we've discussed today, and publishing, of course, review articles on all the topics you've mentioned, both of you today. And so thank you so much again. And I guess I'm going to end with quoting Annie, because I really loved it, with, anything is possible. And I hope to interact with you all at some point in the future again on articles. And thank you all so much for joining and for asking questions, and have a wonderful afternoon or evening. Thank you very much.
Thank you.
Everyone. Thank you.
Bye-bye.
Bye. Thank you.
Bye-bye. Thanks a lot.